Okay, this is chapter 16 on molecular genetics. This is where we talk about DNA. This thing going here. DNA is the genetic material. The role of DNA in heredity stemmed from work from bacteria and viruses. The work of Frederick Griffith. Studying bacteria that cause pneumonia, he found that two strains, he had one that was pathogenic and one was not. Pathogenic means it causes disease. When he killed the pathogenic bacteria with heat and mixed the cell remains with living cells, some of the living cells became pathogenic, and this new pathogenicness was inherited by descendants. This was his experiment. So he has these living cells as a control, and they have this pathogenic bacteria, and the result is the mouse dies because it has the disease. So then he has the living cells that are the control, that do not have the disease. They're not pathogenic. So these are non-pathogenic and these are pathogenic. And the mouse is healthy because the bacteria are not disease, do not cause disease. Then what he does is he takes the heat cells, so these are the pathogenic ones, and he kills them with heat, and the mouse stays healthy. Then what he does is he mixes those heat-killed ones that cause the mouse to be healthy, and he mixes those with the living cells that are non-pathogenic, and mouse dies. So whatever it was that was inside these cells had to then get inside those cells. DNA as a genetic material continued. Other evidence was from bacteriophages, viruses that infect bacteria. Alfred Hershey and Martha Chase found that DNA from the virus was injected into bacteria, which causes the bacteria cell to make new copies of the virus. So here they have, this is a bacteriophage. It's a virus that infects bacteria. And this went in, and they noticed that this genetic material went into the cell. And then over here, we've got kind of a similar situation, except you're using a different type. And they figured out that, let's go back a second. The virus was injected into bacteria that causes the bacteria cells to make new copies of the virus. So these are just two experiments out of a lot that helped us to determine that DNA is actually the genetic material, not a protein, not a carbohydrate, not a lipid. It is actually DNA that is the genetic material. You're going to have to, part of your Chapter 16 reading assignment is to go through and read about some of those history, and you're going to be introduced to several individuals. And then in class, we'll play a Kahoot quiz to kind of review that information. So ultimately, there's all these experiments, and this is what we come up with. Here's what we know about the structure of DNA. DNA is made up of nucleotides. Each nucleotide is made up of a nitrogenous base. That could be adenine, thymine, guanine, or cytosine. Hopefully some of this is coming back now from Bio 1. A pentose sugar. Pentose means 5, so it's made up of 5 carbon ring, which is called deoxyribose, and a phosphate group. Chargaff came along, and he found out that within any given species, that the number of A's was always equal to the number of C's. The amount of adenine equaled the amount of thymine, and this amount of cytosine equaled the amount of guanine. And he figured out that between species that varied. Some species had more adenine than others, and some species had more cytosine than others. But he figured out the amounts were always equal. That led us to the understanding that these bases would always pair together, and these bases would always pair together. So here's one nucleotide right here. And this is going to be the base, and it might be thymine or adenine. Here's your phosphate group, and here's your 5 carbon sugar. 1 carbon, 2 carbon, 3 carbon, 4 carbon, 5 carbon. Actually, sorry, 5 carbon up here. That's an oxygen. And this end here, because that 5 carbon is sticking up, this becomes later. That's what we call the 5 carbon or the 5 prime end. Down here, you've got 1 carbon, 2 carbon, 3 carbon, and since this one's sticking down, we call that the 3 prime end, and that becomes important later in the structure of DNA. You'll also notice that thymine is a little 1 ring, and adenine is 2 rings, and then cytosine is 1 ring, and guanine is 2 rings. And in order for DNA to not to be lopsided, A large ring has to pair with a small ring. How do we know which ones pair together? Well, Chargaff figured out the amount is the same. So if the amount is the same, they must be always found together. Rosalind Franklin used X-ray crystallography to see pictures of DNA, and this was her picture of DNA. Oh, sorry. Her picture of DNA right here. Her pictures confirmed for Watson and Crick that DNA was a double helix. So there's two strands and they're twisted together. Rosalind Franklin unfortunately died before the Nobel Prize came out. Otherwise, she probably would have been the forerunner in that. Watson and Crick used her work and they got the credit, but she had passed away, and that's how that went. So Franklin's pictures further noted that DNA structure is uniform in diameter. Remember, that means that it's not lopsided. So two rings can't go with two rings, and one rings with one rings. Otherwise, you'd have a two ring and a two ring, and then a one ring and a one ring, and it would go like this. And it wouldn't be, but for it to be even, that means there's a two ring with a one ring, a two ring with a one ring, and it's always like that. That's what we mean by uniform. Watson and Crick noted for DNA to be uniform, one purine must fit with one pyrimidine. Purine is a two ring, and a pyrimidine is a one ring. A and G are purines, and T and C are pyrimidines. So put that together with Chargaff's rules, and you realize that A and T have to pair together, and C and G must pair together. There you go. That's what I was trying to draw earlier. Not quite as pretty for me. So here you go. You have a two ring with a one ring. Always a purine with a pyrimidine. DNA is also anti-parallel. That means one strand runs 5' to 3', and the other strand runs 3' to 5'. So in this case, the 5' end is sticking up here, and it's sticking up down here. That means that all of these are going in one direction, and all of these are going in the other direction. That's what anti-parallel means. And I believe that is actually it. Because we are on to that was slide 12. And so we are going to finish this up. In class, we're going to do DNA replication, and we're going to cover several slides, and then I will make another video to cover the remaining information about DNA at the end. So here, slides 1 through 12 just covered the structure of DNA. So you need to make sure you understand purines and pyrimidines and A pairs with T, and what Chargaff's rules are, who Rosalind Franklin was, who Watson and Crick were, who Hershey and Chase were, and who, name is blanking on me, Griffith was. You have to understand what anti-parallel means. Other things that you might be interested in knowing is the hydrogen bond. You're going to need to know about the hydrogen bond. So what connects these base pairs to each other is a hydrogen bond. It's kind of weak, which is nice because then it can easily be replicated. You can pull that apart. You have to know that this is a nucleotide, and that this is the phosphate, this is the sugar, and this is the nitrogen base. And there are 4 bases, A, T, G, and C. And A and T go together, and C and G go together. You have to understand that this is what the 5' end means. That means that's the 5th carbon sticking up there. And over here, it's going to be the 3rd carbon that is sticking down there. I'm trying to think of what else. The backbone, this would be what we consider the backbone. It's made up of alternating phosphates and sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. And then these are going to be your nitrogenous bases. I think that might be it. Okay, hopefully this is a review. I don't think I really added much since we did this in Biology 1 other than talking about the anti-parallel.